Thyroid and Parathyroid Glands, Part 2. In this video, you'll be introduced to some of the concepts that are listed here on this slide. We'll also talk in more detail and discuss the interactions of some of these metabolic and physiological reactions in the classroom. But we'll introduce here the three major factors in controlling calcium homeostasis, particularly the concentration of calcium in blood. We'll be able to discuss the mechanisms controlling the release and actions of the PTH. We'll be able to describe the control of calcitonin release and how PTH and calcitonin play with each other in an antagonistic kind of way. We'll discuss the synthesis of the active form of vitamin D3 and what is it that it does. We'll also talk about interactions that affect and have effects of these three hormones and all the other peripheral tissues and visceral organs that play a part in this interaction, controlling both calcium and phosphate levels. Now we're going to talk about three hormones and how those three hormones affect the concentration of calcium and phosphate. Before we consider calcium and phosphate independently, we need to keep in mind that when calcium and phosphate are together, we deposit salts. Calcium phosphate is an ingredient that is necessary for bone deposition. So if you're out of bone tissue, you definitely want calcium and phosphate to be inversely proportioned to one another, meaning if there's a lot of calcium, there shouldn't be enough phosphate. Otherwise, then you get a deposition of crystals or salts. Calcium on its own, we know that is involved in muscle contraction, myofibril contractions. We also know that when calcium enters a presynaptic neuron, synaptic vesicles containing neurotransmitters will be facilitated to be fused in the presynaptic membrane. We also know that calcium plays a role in the excitability of cell membranes in general, and it also plays a part in enzymatic activity. Phosphate, in turn, has a role in metabolism. We know that phosphate is one of the components of high-energy molecules that are used for as fuel for cellular processes such as ATP. We also know that some of the enzymes get activated once a phosphate group attached to them. That process is called phosphorylation. We also know that phosphate is stored in bones as well. I should probably also mention that calcium is stored in its greater majority in bone tissue. The three hormones that regulate the concentration of these minerals are PTH, secreted by the parathyroid gland, calcitonin, secreted by C cells or parafollicular cells found in the thyroid gland, and finally vitamin D3. Vitamin D3 synthesis is a multi-step process, and I will talk about that in subsequent slides. Calcium is stored mostly in bone tissue, but we do know that only a small fraction of calcium found in bone is actually active under hormonal regulation. Calcium is also excreted through urine via the kidneys. We also know that the kidneys are subjected to hormonal regulation too. Finally, through dietary intake, we get our calcium, and it's being reabsorbed in the gastrointestinal tract. The way we synthesize PTH in the parathyroid gland is through a pro-parathyroid that gets sliced to form a pro-parathyroid that also gets modified. And finally, we get the last product, the parathyroid hormone. We also know that low concentrations of calcium in blood will release the majority of the PTH in this gland. However, there are some sensors in the chief cells within the parathyroid gland that can sense differences in concentration of calcium in blood, and that could also trigger its secretion. In terms of physiological effects, we know that from previous slides that calcium and phosphate are usually opposite in concentration if we're talking about tissues that are out of bone. So in the kidneys, they have the exact opposite effect. We preserve the calcium in the kidneys, so excretion is decreased, reabsorption is increased, and the opposite thing happens with phosphate. PTH will also facilitate the reabsorption of calcium in the digestive tract as long as vitamin D3 is present. Also, it mobilizes calcium from bone. Remember, bone is the primary storage for calcium. And it does that, PTH affects that by the presence of another mediator called interleukin 1. On this slide, you can see the negative feedback loop for hormonal regulation of PTH. We know that if there's a drop in plasma calcium concentration, that is going to trigger a series of reactions in which we will maximize and increase the intestinal calcium absorption at the GI, and we will go to our storage units, bone tissue, to remove some of that calcium stored there to make it available in the bloodstream. There's also a lot of other ways that we can excrete excess of calcium, mostly through the urine and the gut. We can also store if it's in excessive amounts, and also it can be exported to the fetus in a pregnant woman, or as part of milk production in the mammary glands. So now we're going to move on to vitamin D. Vitamin D is an interesting, again, multi-step process in which it's being synthesized. So basically what you have, after exposure of sunlight in the skin, there is a conversion from 7-dehydrocholesterol to cholecalciferol, and that's vitamin D3. That's one way of getting it. You can also get vitamin D3 by dietary intake. Vitamin D3 is a nutritional supplement that's high in concentrations in fish and meats, and you can also take it as supplements, usually in the form of vitamin D2. Now, in the liver, vitamin D3, it's converted to another intermediate component called 25-hydroxyvitamin D3, and we call that calcidiol. Calcidiol then will be converted into calcitriol with a T in the kidneys. Another name for calcitriol is 1,25-dehydroxyvitamin D3. Now, this is the active form of the hormone. We also know, and we'll explore this more in class, that calcitriol is also affected or regulated by concentration of PTH, sex hormones, growth hormone, and even insulin. The physiological effects of vitamin D3 are quite interesting. This will activate the calcitriol receptors in these three places: the duodenum, osteoblasts, and kidneys. And eventually, it facilitates the release of an enzyme called alkaline phosphatase. This enzyme will suppress the inhibitors of bone mineralization. It's almost like a double negative, which makes it a positive. So the effects of the release of this enzyme is going to facilitate mineralization of bone. It's going to make bone stronger. Now, we need two ingredients for bone, as we said before, and the ingredients that we have mentioned are calcium and phosphate. So in the GI, vitamin D is going to facilitate both the reabsorption of calcium and phosphorus, and same thing in the kidneys is going to facilitate the reabsorption of both calcium and phosphate. And finally, we'll mention a brief function of calcitonin. We know calcitonin, again, was synthesized in the parathyroid gland, and the actual end form of calcitonin is or comes from a very, very modified version of a procalcitonin that has been sliced and again modified in a couple of different steps. The release of calcitonin, and what facilitates it, is an increase of calcium levels in blood. However, calcitonin is not actually required to keep homeostatic levels of calcium in blood. This chart shows multiple steps and processes. I'm only interested in highlighting the top part of it, where calcitonin has a direct effect on osteoclasts by inhibiting them. That, in turn, will stimulate calcium deposition in bones, which is really what we want. Calcitonin is only released if there's a peak or an increased concentration of calcium in blood, so we probably have more than what we need. What do we do with the excess of calcium? We want to store it, and the place to store it is mostly bone. So calcitonin helps in doing that. It also helps in the inhibition of actually more bone reabsorption by the osteoclasts, which make perfect sense that it would inhibit these cells that do that. This chart summarizes the effects of PTH, vitamin D3, and calcitonin in the plasma concentrations of both calcium and phosphate. It also shows what happens in terms of excretion of these two minerals. But for now, let's just focus on the plasma concentrations of both calcium and phosphate. PTH, after it's secreted, will facilitate an increase in plasma calcium. The antagonistic calcitonin will do the exact opposite. Vitamin D3 serves as a synergistic hormone with PTH. In terms of phosphate, PTH is going to decrease phosphate concentration in blood. Calcitonin, in this case, is a synergistic hormone for phosphate. The antagonistic is vitamin D3. When it comes to calcium excretion through urine and phosphate excretion, PTH do the opposite for both calcium and phosphate. So PTH will decrease urine calcium while increasing phosphate urine. Vitamin D3 is going to do exactly the same for urine calcium or excretion of calcium through urine, but is going to do the opposite for phosphate urine. Calcitonin has no effect on the kidney. In class, we'll talk about how these three hormones interact with each other and how dysfunctions may have a potential impact on all these effector organs, but mostly bone tissue.